each cell of the sporogenous tissue that is there, it will form what? Microspore tetrads. The formation of embryo sac from a single megaspore is called as monosporic development. Nucleus of the functional megaspore, again that will undergo what? Mitotic division to form two nuclei. Microsporogenesis is nothing but the formation of the pollen grains from the pollen mother cell by reduction division or by meiosis. Hello everyone, a warm welcome to this session on class 12th biology for CBSE. I'm Dr. Divya, biology faculty. So in this session, we'll do the continuation of session 2, that is of chapter 1, sexual reproduction in flowering plants. So in the previous session, we had dealt with the male reproductive structures. In this session, we'll deal about the pre-fertilization events that occur, meaning before fertilization, what all happens between the male and the female reproductive structures of the flowering plant. Learn about the pre-fertilization events, that is microsporogenesis. So if you remember, in session 1 of the same chapter, we had dealt with the microsporangium, that is the structure of the anther. Inside, when you take a TS of the anther, inside the anther, we could find the microsporangium. We learned the structure of the microsporangium, wherein it is made up of 3 layers, or it is covered by 3 layers of cell, that is the epidermis, then comes the middle layers and the endothecium, and within the endothecium there is a nourishing layer which is called as the tapetum. And also we studied about the microspores, right, or microspore mother cells. Now from these microspores, which are nothing but the pollen grains, how exactly do they get produced? Or the genesis or the formation of the microspores or the pollen grains is called as the microsporogenesis. So the pollen grains need to be formed and they need to get released, no, if the fertilization events has to occur. Continue. So therefore, before fertilization, that is before the fusion of the male and the female gamete, what all happens is what we are looking into. So microsporogenesis is nothing but the formation of the microspores, or we can say it is the formation of the pollen grains, because the pollen grains are nothing but the microspores. Microspores from a pollen mother cell, that is the pollen mother cell. Some of the cells, that is some of the pollen mother cells, they undergo reduction division, which is called as meiosis, and during that process pollen grains get produced. So that is called as microsporogenesis. Micro, minute, sporogenesis, formation of the spores. What type of spores here? Microspores. So therefore, microsporogenesis is nothing but the formation of the pollen grains from the pollen mother cell by reduction division or by meiosis. Now, what are the steps involved in the microsporogenesis or the formation of the pollen grains we will look into? So the first step is the anthers that are there. So what is the anther? It is the male reproductive part of the flower, that is it belongs to the stamen, right? So in the stamen we have the filament which is attached to the anther. Now that anther, which is the bilobed structure, they actually dehisce to release the pollen grains. Now before that the pollen grains need to be produced. So when the anther develops, the cells of the sporogenous tissue. Sporogenous tissues means those which give rise to the spores are called as the sporogenous tissues. Those sporogenous tissues, wherein in that the pollen mother cells that are there, they will undergo reduction division to form microspore tetrads or pollen tetrads. Tetra means 4 in number. So what will happen? They'll undergo reduction division to form microspore. Now this is a tetrad. Microspore tetrad. Why? Because here there are 4 spores that are developed, right? So it is a microspore tetrad and it is N. It is haploid. Why? Because it is a reduction division. After the formation of microspore tetrad, what will happen? We will look into. Now, each cell of the sporogenous tissue that is there, it will form what? Microspore tetrad. So where is the sporogenous tissue present? Inside the microsporangia, right? So these microspore tetrads that are there, among lots of microspore tetrads that are formed, once the microspore tetrads are formed, the anther will mature. So as and when the formation of the microspore tetrad is occurring, the anther will not be able to hold it because of the pressure. It needs to release, or it needs to dehisce or split open to release the pollen grain. So as and when the anther matures, the layers that are there, that is the microsporangia layers that are there, they will become dry, and because of the dryness and because of the increasing number of the microspore tetrads that are there, or the pollen grains that are there, they will just burst open, and or the microspores, after that, once the anther is matured, what happened? The microspore tetrads that were there, they will dissociate from each other. This is what will happen. They will get released. Now, it will no longer be called as a tetrad. This is actually a microspore tetrad, but they will leave one another. That is, they will disintegrate from one another, and they will form a single cell, separate cells. And these cells are nothing but the pollen grains. And when the anthers, they split open, or when they dehisce, thousands of microspores or the pollen grains are released. So that is how the pollen grains are released. So now we know about microsporogenesis. So got to know how from the anther the pollen grains are released. Now the pollen grains are released and they are ready for fertilization to occur. So now we know about the anther. So we now need to know about the formation of the megaspore, right? That is the ovules, how they get developed is what we need to know here, because we are studying about the female reproductive structure now. So that is called as megasporogenesis. That is the formation of the megaspore. So what is megasporogenesis? It is the process of formation of megaspores from the megaspore mother cell. And the steps involved in megasporogenesis is, here also the megaspore mother cell that is there, it will also undergo reduction division, that is meiotic division, and it will produce 4 megaspores. Same way, in the pollen grain, that is in the microspores, what happens? The microspore mother cell or the pollen mother cell, they will undergo reduction division or meiotic division to form a tetrad, that is a microspore tetrad, in which 4 pollens will be present, right? Therefore, it is called as a tetrad. Same way here, the megaspore mother cell will undergo reduction division or meiosis to produce 4 megaspores. If we can see here, this is the megaspore mother cell. They will undergo reduction division to produce how many? 4 megaspores. So this is 4 megaspores that they produce. So out of the 4 megaspores, all the 4 will not function. In that, only one will be generative and the other or one will be functional. Out of the 4 megaspores, how many? Only one is functional and the other 3 that are there, they will get degenerated. They are of no use. So therefore, only one megaspore is functional. So that one megaspore which is functional, it will develop into the female gametophyte, embryo sac. Remember, we had studied the structure of the embryo sac in the previous session. You can go back to a previous session and have a look to it. So this is nothing but the embryo sac. From where did this embryo sac form? It formed from the megaspore mother cell. So the megaspore mother cell present where? In the ovary, right? So in the ovary, in the ovule, what happens? The megaspore mother cells that are there, they will undergo meiotic division or reduction division to form 4 megaspores. Out of the 4 megaspores, only one is functional. That one which is functional will later on develop into the embryo sac, because now the embryo sac is also mature. The matured pollen grains are also released by the anther. Now the only thing that has to happen is fertilization. That is the fusion of the egg that is present in the embryo sac with that of the pollen, male gamete present in the pollen grain. So that is nothing but the megasporogenesis here. So monosporic development. So there is a term given in the textbook. What is monosporic development? Mono meaning one, right? Mono meaning it is one. So monosporic is nothing but the formation of embryo sac from a single megaspore is called as monosporic development. That's true, right? Because only one that is functional will develop into a megaspore. So, will develop into the embryo sac. So, therefore, the method of formation of embryo sac from a single megaspore is called as monosporic development. And if we brush up, we already studied what is there inside the embryo sac. To again brush up, inside the embryo sac, they have the central cell. In the central cell, what it is made up of? It is made up of two polar nuclei, and there are two synergids in the micropylar end and one egg cell is there, and here three antipodal cells will be present. So this is the structure. I just labeled it again so that for the fertilization next session it is very important. So this is about the structure. Next we'll study about the formation of the embryo sac. So what did we get to know? That one of the functional megaspore will develop into the female gametophyte, which is the embryo sac. Now exactly how the embryo sac formation will occur, we will look into. So the steps involved in the formation of embryo sac. Because we got to know embryo sac will get formed. Inside the embryo sac, cells and all started to develop. From where did these cells come? That is what we need to learn now. Like we have the synergids, the antipodals, the egg apparatus, or the egg apparatus wherein synergid is there, egg cell is there. Then towards the chalazal end, three antipodal cells were there. Then in the center, one central cell is there, inside which two polar nuclei are there. From where did all that come from? That is what we will learn here. That is the steps involved in the formation of the embryo sac. So now inside the megaspore, that is one of the functional megaspore. We learned, right? We had learned that out of the 4 megaspores that is formed, only one is functional. That functional megaspore will develop into the embryo sac is what we got to know. Now inside that megaspore, it is a cell. So obviously it will have a nucleus, right? So the nucleus of the functional megaspore, again that will undergo what? Mitotic division to form 2 nuclei. How many? 2 nuclei will get formed, because mitosis is nothing but equational division. It will split into equal halves wherein 2 nuclei will get formed here. Now the 2 nuclei will move to the opposite poles and form the 2 nucleate embryo sac. First, what will happen in the first step? 2 nucleate embryo sac will get. So this is the 2 nucleate. I'll write it as ES, okay? ES is nothing but embryo sac. So what is this 2 nucleate embryo sac? As I told you, the functional megaspore, it had 1 nucleus, right? So that 1 nucleus will undergo mitotic division, that is equational division, and it will form 2 nucleus. That nucleus is in the opposite poles. On the opposite poles they will be present like this. They will move to the opposite poles. Now these 2 nuclei which are there inside the embryo sac in the opposite pole will again divide mitotically. Therefore, when they divide mitotically, how much nucleus will get formed? 4 nucleus. So can you see here? This will divide. Again, when it divides, what happens? It will form 2 nucleus. This will divide to form 2 nucleus. Therefore, how many nucleus inside this? 4 nucleus. Therefore, it will be a 4 nucleate embryo sac will get formed. So this is a 4 nucleate embryo sac will get formed. Now again those 4 nuclei which is there, again it will divide mitotically to form 8 nucleate embryo sac. It will form 8 nucleate embryo sac. Now here 8 nucleus is there. So therefore how many cells it can form? 8 cells, right? Why 8 cells? Because in each of the cell 1 nucleus has to be there. But here exactly only 7 cells will get formed. We had studied in the previous session. Embryo sac is 8 nucleate but still it is 7 celled. Why it is like that? It is because after the 8 nucleate stage the cell walls will develop. So will the cell wall develop in around all the nucleus? No. Only around 6 nucleus it will develop. Cell wall will get formed. Now how many cells got formed? 6 cells gets formed. So out of the 8 nuclei, 6 are surrounded by the cell walls and they get organized into cells. Now what will happen to the next? the remaining that is there. So as I told you, the remaining. So see here, the cell wall will get formed. So this is the cell wall formed around them. So 2 will be remaining. So 2, whichever the nucleus which was not covered by the cell wall, they'll just move towards the center and they will settle here and they will develop a cell wall around them. So now inside the cell wall, how many nucleus is there? 2 nucleus, right? So therefore, how many cells will get formed here? 1, 2, 3, 4, 5, 6, 7. 7 celled, each having one nucleus. But the central cell that is there, it will have 2 nucleus. Therefore, it is 7-celled, 8-nucleate stage. Okay. So that is how the embryo sac will develop. So the remaining 2 nuclei, what will happen, as I told you, it will move to the center and they are called as the polar nuclei. So this is nothing but the polar nuclei. And the 3 cells, they get grouped together at the micropylar end. Now this is the micropylar end and this is the chalazal end. Now in the micropylar end, 2 cells, they form the synergids. 1 cell will form the egg apparatus and the remaining cells which moved towards the chalazal end, it will form the antipodal cells. So this is how the embryo sac will develop. Now we had learned the structure of the embryo sac in the previous session. How the embryo sacs, inside the embryo sacs, all these cells developed, we got to know that, right? It was because of the mitotic division of the nuclei that was present in the megaspore mother cell. That is one of the functional megaspore mother cell. So this is about the pre-fertilization events. So in this session, we got to know about how the pollen grains are developed and how they get dehisced from the anther. Now the pollen grains are ready, and in the second session we got to know what all are the components that are present inside the pollen grain. That also we got to know, and we also got to know about the embryo sac. Inside the embryo sac, how the formation of the cells and all take place, we got to know, and we got to know that it is a 7-celled 8-nucleate stage. All that we know now. So we know the structure of the pollen grain, the embryo sac. So in the next session, it will be easy for us to learn about the post-fertilization or the pre-fertilization events itself, right? So in the pre-fertilization events, now the pollen grains have come out of the anther. So what will happen to the pollen grains? They have to go and sit on the stigma. That particular process is done by pollination. So what is pollination? What are the different kinds of pollination and what are the agents of pollination is what we'll study in the next session. So I hope you found this session interesting and it was useful for you. So see you in the next session discussing about the topic pollination under the same chapter. So see you in the next session. Thank you.